So today we're going to talk about aldolase gene expression. My name is Katie Ochoa, and this is for a course I'm taking at Grand Canyon University, Chemistry 530. And today is actually December 13th, 2020, but you'll see the assignment due date in the lower left-hand corner. Let's get started talking about aldolase gene expression. After today's lesson, you should have an understanding of glycolysis and gluconeogenesis. We're going to start with an overview of both of those processes. And you'll see that the aldolase enzyme plays a role in both processes. It's part of a reversible reaction that is a step in both the breakdown of glucose and the formation of the 6-carbon molecule of glucose. There are 3 different isoforms of aldolase that you will be introduced to during today's lesson. We'll talk about where those different forms are expressed, and please keep in mind that today's discussion is focused on aldolase expression in mammals, but the aldolase enzyme has the same activity even in protists, fungi, bacteria, in all cells in which it is present. And then we're going to end the discussion with how aldolase B, which is one of the isoforms or isozymes of aldolase, how it can be regulated by transcription factors. And we're going to talk about 4 different transcription factors: HNF1, HNF3, CEPB, and DBP. So remember these objectives as we move through today's lesson. Let's go ahead and get started. Let's remind ourselves of what glycolysis is. It's a process through which glucose is broken down in the cell, and it's a series of 10 reactions. Glycolysis yields 2 molecules of pyruvate, which is a 3-carbon compound, from glucose, and also results in a net yield of 2 ATP and 2 NADH, which are key for electron transport and generating much more ATP in the mitochondria of the cell. So glycolysis is important for its NADH production, not as much as its ATP production. I'm sorry, the NADH production is much more important than the 2 ATP produced by glycolysis. Here we have the reaction that occurs in the glycolysis cycle. Glucose plus 2 NADs and 2 adenine diphosphates plus 2 phosphorus results in the formation of 2 3-carbon pyruvates, 2 NADH, which will be transported to the area of the mitochondria where electron transport occurs, 2 hydrogen, 2 ATP, and 2 molecules of water. And this is per glucose molecule. Okay, here is an amazing diagram of the glycolysis process. And I just want to take a second to talk about each of the steps and then point out that we will be focusing on this enzyme right here involved in step 4 of the glycolysis process and also involved in the reverse process, which is called gluconeogenesis. So step 1, hexokinase. This glucose ring, which has 6 carbons, is phosphorylated by ATP into I'm sorry, I lost my place. It catalyzes the phosphorylation, and then we get this glucose 6-phosphate. I got ahead of myself and was talking about the 6-carbon ring that's turned into the 5-carbon fructose 6-phosphate in this second step. And the enzyme responsible for that second step is phosphoglucose isomerase. Step 3, phosphofructokinase, which uses magnesium as a cofactor and also consumes another ATP, adds another phosphate, which is why we become a bisphosphate, to the fructose molecule. So now we have fructose 1,6-bisphosphate. Okay, here's the step that we're concerned with. This aldolase enzyme splits the fructose 1,6-bisphosphate into 3 different 3-carbon compounds. The first, dihydroxyacetone phosphate, must be further converted by another enzyme, this triose phosphate isomerase, into something that can continue in the glycolysis pathway. And the other 3-carbon product of aldolase is glyceraldehyde 3-phosphate. This is the only one of the 2 molecules created that continues in the glycolysis cycle. So this other dihydroxyacetone phosphate must undergo another reaction in order to continue in the glycolysis pathway. So this DHAP is reorganized by this enzyme right here into something that can continue in glycolysis. Let's see. In this step right here, we have a phosphate, which does not come from ATP this time. So this does not consume another ATP. That the phosphate is added to this GLAP, the GAP, glyceraldehyde phosphate, and we have the creation of 1,3-bisphosphate glycerate, and we also get an NADH and a hydrogen, which are both electron carriers, key for energy production after glycolysis. Let's see. After we have 1,3-bisphosphate glycerate, in comes this kinase right here that we actually get the production of an ATP. Since we have, let's see. So now we're balancing out the use of one of the No, we've balanced out actually the use of all of the ATP so far because we have 2 3-carbon compounds that are continuing through the glycolysis pathway. So this actually we get the production of 2 ATPs as each 3-carbon compound now is moving through glycolysis. Right here, this phosphoglyceromutase simply moves the phosphate group from the 3 to the 2 position. In step 9, enolase removes a water molecule. And then in 10, the pyruvate kinase, we have the production of another 2 molecules of ATP, which is what yields our net overall production of 2 ATP molecules. And we also get the final product of glycolysis, which is 2 3-carbon pyruvate molecules. So this is glycolysis, an overview of glycolysis. And again, I'd like to remind you that we are going to concern ourselves very specifically with the aldolase enzyme involved in step number 4, where we take fructose 1,6-bisphosphate and convert it into these 2 3-carbon products, DHAP and GAP. Okay, so gluconeogenesis, which is kind of the opposite of glycolysis. Rather than breaking down the six carbon glucose, gluconeogenesis takes non-carbohydrate sources of carbon and creates glucose molecules. This occurs when glucose is unavailable. So the cell creates glucose and catalyzes it from non-carbohydrate sources, such as lactate, pyruvate, glycerol, or certain amino acids. Here is the reaction for gluconeogenesis: 2-pyruvate, and we do need 4 ATP, adenine triphosphate, and 2 GTPs. So we need more energy for gluconeogenesis than we do glycolysis. We also need 2 NADH and 6 water. And this process results in glucose, 4 adenine diphosphates, 2 GTPs, plus 6 phosphates, 2 NAD plus, and 2 protons. So, in order, like I said, to make this reaction energetically favorable, we do need to use a lot more energy. And it's not exactly the reverse of glycolysis. In glycolysis, 7 of the 10 steps are reversible, and these 7 steps are the ones that are the same as gluconeogenesis, but occur obviously in the opposite direction. 3 of the steps, though, are essentially irreversible in glycolysis, and these are the ones that differ between glycolysis and gluconeogenesis. And the 3 steps that are different between the two processes are let me first point out that this pathway right here in red is the glycolysis pathway, and then here we have gluconeogenesis. So what's highlighted here are the enzymes used in the different reactions between gluconeogenesis and glycolysis. And here you'll see that we are moving this direction and breaking down glucose into pyruvate. And in this process, the gluconeogenesis, we're taking pyruvate and working backwards to make glucose. The first difference in the two processes are where pyruvate kinase is used to go from phosphoenol pyruvate to pyruvate. In order to reverse that reaction, we actually need 2 different enzymes, the pyruvate carboxylase and PEP carboxykinase, and it has to do with oxaloacetate's inability to move across a membrane. So we actually have to do this in 2 steps. We have to change it into something that allows this process to occur. The other difference is going in this direction in glycolysis, we use phosphofructokinase, or the cell uses phosphofructokinase 1 to make this conversion. And in the reverse, the enzyme that is used is fructose 1,6-bisphosphatase. And then here we have our third difference in enzymes. In glycolysis, we use hexokinase. And in gluconeogenesis, the enzyme that changes glucose 6-phosphate to glucose is glucose 6-phosphatase, which of course removes this phosphate group from this molecule so that we get glucose produced in the cell. I think that's it for this slide. Let's go ahead and move on. Okay, so in glycolysis, aldolase is responsible for the generation of dihydroxyacetone phosphate, DHAP, and glyceraldehyde 3-phosphate, or GAP. In gluconeogenesis, aldolase is, of course, responsible for the opposite. So it takes these 3 carbon molecules, the gap and the DHAP, and it turns them into 1,6-bisphosphate on the pathway to producing a glucose molecule. So why do we even have a gluconeogenesis pathway? And why is it important? And in fact, it's so important that all species can synthesize glucose using this pathway. And that really indicates its evolutionary importance. And all cells, not all cells, all organisms have an ability to complete gluconeogenesis because early on in evolutionary history, there was a need to synthesize glucose. Glucose molecules were not readily available in the biosphere. So before there was an accumulation of glucose, all organisms needed the ability to produce their own glucose. And then when it was more readily available in the biosphere, there needed to be a process to recover the energy that cells had to expend in order to produce their own glucose. So that's when glycolysis began to evolve in the cells. But still, overall, gluconeogenesis is more important than glycolysis. Perhaps not in mammals, but when you consider plants and think that plants, their only source of glucose is their production of it from carbon dioxide, you can understand how important gluconeogenesis is, though it doesn't seem to be so since we as mammals consume all of our glucose, and glycolysis is a more important process for us. Okay, so let's continue our focus on aldolase. And let me just refresh because I feel like I went off on a tangent there trying to explain the importance of gluconeogenesis. But just to refresh that aldolase is responsible for taking the 1,6-bisphosphate, and this is a reversible reaction between turning 1,6-bisphosphate into dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. So it takes 2 3-carbon compounds and turns them into a 6-carbon compound, or the reverse in glycolysis and gluconeogenesis. There are actually, and we're going to focus on mammals now, of this enzyme, there are 3 isoforms of the aldolases, and class 1 aldolases occur in mammals. There's 2 classes, and I want to say that the other class is the one that occurs in protists, bacteria, fungi. But let's focus on aldolases that occur in mammals. So the A isoform is expressed mainly in the muscle. It localizes to the cytoplasm, and it's proposed that high levels of aldose A in tumor cells is actually indicates a very poor prognosis because it's thought to promote glycolysis in tumor cells, which results in growth and proliferation of the cancer cells. The B isoform, which we are actually going to look at a bit more in depth, or rather expression of the B isoform, is expressed in the liver, kidney, stomach, and intestine. It's found in the cytoplasm and perinuclear membrane of hepatocytes. And an increase in dietary carbs has been found to lead to an increase in aldolase B gene transcription in animal livers. Just a note to consider. The C isoform, the C isoform is expressed in the brain, heart, and ovary. And we're going to go ahead and look at the B isoform and look at some patterns in its expression and how it is regulated. But before we do that, so these three isoforms of aldolase, they are very similar in sequence. Actually, A and B are 66% similar. B and C, 78% similar, and 78% similarity in the identity of aldolases A and C. So very similar genetic sequences for the aldolases, and that of course can be linked to very similar action for all aldolase isoforms, though they are expressed in different tissues. In adult hepatocytes, aldolase B is the only aldolase isozyme gene expressed. So you are not going to find aldolase A or C in the liver tissue. So when trying to determine how aldolase B expression was regulated, Gregory, Kahn, and Pitchard looked at several factors known for controlling liver-specific transcription. So they tested all of these factors to see if they would affect the transcription of the aldolase B gene, which I've referred to throughout the rest of the presentation sometimes as ALDO-B. So if you see that, I'm talking about the aldolase B gene. And these researchers found that HNF1, DBP in hepatoma cells, and CEBP in hepatocytes are major upregulators of aldolase B gene expression, and HNF3 is a strong inhibitor of aldolase gene expression, and it was found to competitively interact or counteract HNF1, which is, like we said, an upregulator, and actually non-competitively counteracts, I believe it's the CEBP factor. So let's talk a little bit about the ALDO-B gene. There's an activating region inside the first intron and then a proximal tissue-specific promoter. So this tissue-specific promoter, which is, of course, specific to the liver, because that's where aldolase B is being expressed, is why several liver-specific factors were tested. All of these liver-specific factors were tested because of this region in the aldolase B gene. They wanted to test the effects of all of these liver-specific factors, or these factors that were known to act on liver-specific tissues, to see if they would in fact affect the ALDO-B gene expression. So what they found with CEBP and DBP. So CEBP forms heterodimers. DBP does not. And they found that these two factors were specific to hepatoma and hepatocytes. DBP was an activator in hepatoma, but actually had no effect in hepatocytes, and CEBP was actually the opposite, efficient at activating the ALDO-B gene expression in hepatocytes, but not hepatoma. So it was thought that perhaps it was because of the structure, as CEBP is able to form heterodimers, while DBP is not. But they found that while it is due to the structure and the activation of the promoter region caused by the structure, it's not due to exclusively this factor or the binding to a receptor, but also the binding of other factors on neighboring sites of the gene that are actually dependent on the cell, hepatoma or hepatocyte, that also affect how DBP and CEBP, if they are able to upregulate the expression of aldolase B in the cell. So then they looked at also HNF1 and HNF3, which were also found to have an effect on aldolase B gene expression. And they found that actually both of these factors bind the same region of the aldolase B gene. The sites, I apologize, were actually, oh my goodness, overlapping. So essentially, the gene could bind the HNF1 or the HNF3. And HNF1 is a very strong activator. HNF3 is not. But it was found that they competitively bind, and HNF3 actually binds much more strongly than HNF1. And since they're both competing for binding sites, HNF3 is the one that binds more strongly and is not as strong at promoting the expression of the ALDO-B gene. So that's why its competitive binding with HNF1 is what creates the condition of HNF3 kind of being an inhibitor of ALDO-B gene expression. So in order to promote the expression of ALDO-B, HNF1 has to be present in higher concentrations, so then it is more likely to bind the promoter region and incur the expression of the ALDO-B gene. When there is a HNF1 present, it can then heterodimerize with the CEBP, and that also causes the expression of the ALDO-B in the hepatocytes. So this also explains why HNF3 has an effect on the CEBP upregulation of ALDO-B gene expression. It's a non-competitive effect, as it's the effect of HNF3 on CEBP is kind of secondary. It's more caused by its effect on the HNF1. So I would just like to close the lesson by reminding you of what you should have learned from this. So I'm just going to go ahead and go back to the objectives to close it up. So you should have an overview of glycolysis and gluconeogenesis, An idea of the role of aldolase in both processes. Quickly, it's the conversion of the 6-carbon glucose to 3-carbon compounds and the reversible process. So in gluconeogenesis, it's going from 3 carbons to a 6-carbon compound. And in glycolysis, the reverse is true. We talked about the different isoforms of aldolase, isoform A, B, and C, where they are expressed in the body. And then we focused in on aldolase B and its expression in the liver, and how that expression can be regulated by these 4 transcription factors: HNF1, CEPB, and DBP all they all promote ALDO-B gene expression. HNF3 inhibits it, and it competes with HNF1, and it has a non-competitive inhibition effect on CEBP. And of course, I owe all of this information to the following sources. Thank you and have a good day.